Okay, so good afternoon, everyone. I'm Mark Schumacher, and I'm helping to launch this, our first PARC seminar. And just before we introduce Dr. Nigel Bunnett, I just wanted to lay out just a couple exciting things that are happening. So PARC has kind of become more than an idea. It's the Pain and Addiction Research Center. And it's an attempt to bring people together to collaborate across campus, both in the pain and the addiction field. And we have a website up and running, and we're certainly interested in you not only participating in the seminars, we have some other activities coming up. I put the QR code there if you'd like to check out the website, in particular our membership efforts. One of the, I think, most important parts is that we are forming essentially scientific working groups in a number of thematic areas that include addressing disparities in care for pain and addiction, as well as our hope to identify new ways to treat and prevent pain and addiction, as well as educational efforts, work in the area of pediatric pain and addiction, as well as a focus on cancer pain. So please check out our website and stay connected. Just briefly, a few dates coming up. Our next kind of event will in fact be what we'll call a PARC retreat, again, all virtual. This will be October 19th. And as well as then the next following PARC seminar, which will have the theme of addiction and reward. And finally, looking way ahead, we are actually planned an international meeting, which unfortunately was originally going to be in person, but I think it'll likely be virtual still in March of next year. So save the date, March 13th. So it's a real pleasure to introduce Nigel Bunnett. I had the pleasure of collaborating with him as well as another colleague, Helga Eilers, here at UCSF. Now it's been, I guess, 10, 11 years ago when we worked on volatile anesthetic effects on TRPA1 and sensory neurons. But it looks like, as you can see there, Nigel has quite a global outreach. And again, we just want to recognize him as family, having been at UCSF and interacted with so many of us. So again, it's my great pleasure to welcome Nigel, and really interested in this exciting work that he has initiated. So with that, I'm going to stop sharing and then have Nigel take over at this point. I might add that Alan Nussbaum is going to moderate some questions toward the end. Thank you. Oh, and finally, please mute, if you're not already, please mute your microphone.
Thank you, Mark. Can I just check that you can hear me okay?
Yes, loud and clear.
Thank you. Well, thank you very much for this kind invitation. It's wonderful to be back at UCSF, if only in spirit, to discuss the hypothesis that endosomes are platforms for signaling pain. This is an image of a spinal neuron, and in green is the neurokinin 1 or substance P receptor, which is massively internalized after a painful stimulus. Of course, I don't need to explain to this audience that pain is an evolutionary conserved mechanism that's required for survival because it allows the avoidance of danger and the awareness of injury. Its importance is illustrated by the sometimes fatal injuries sustained by individuals with a congenital insensitivity to detect pain. Normally, pain is tightly regulated and acute, but if it becomes dysregulated, it can give rise to chronic inflammatory and neuropathic pain, which can afflict 1 in 5 people at some point in their lives. The mechanisms that underlie the transition between acute and chronic pain are poorly understood, and in consequence, current therapies are inadequate, as illustrated by the opioid crisis, responsible for over 70,000 deaths per year in the US alone. My lab studies the role of G-protein coupled receptors in pain transmission. These are 7 transmembrane domain proteins which couple to heterotrimeric G proteins. With almost 1,000 members in humans, this is the largest family of receptor molecules. They regulate most physiological and pathological processes and are the target of over 1/3 of FDA-approved drugs. we study the function of these receptors in the first cells in the pathway to pain. These are primary sensory neurons, which have cell bodies in dorsal root and trigeminal ganglia, and which project nerve fibers to peripheral tissues, such as the skin, and to the dorsal horn of the spinal cord, and second-order spinal neurons that transmit information centrally. The peripheral projections of primary sensory neurons are equipped with a large number of G-protein coupled receptors, receptor tyrosine kinases, and TRP channels, allowing them to detect structurally diverse ligands that are generated in injured tissue. Upon activation, a subset of these neurons release calcitonin gene-related peptide and substance P, which activate their G-protein coupled receptors in the vasculature to cause neurogenic inflammation.
Excuse me, Nigel. Sorry, you're showing us the preview, I think, of your slides. I'm not sure we're in the full mode for everyone. Is that right, Anna? Sorry to interrupt.
Okay,
If not, we'll just continue. I just wanted to make sure that you were aware that it might
can
be.
you see? Yeah, that's. You can actually see my slide though. Is that right?
Yes.
I think the problem, Nigel, is you might be sharing your desktop instead of the actual. So that's why we're seeing everything, perhaps. But I think you really want to try to get just the image because it'll be bigger.
I do, and let me stop sharing for a second. Let me start sharing again. Hang on, let's see. Desktop. Actually, let's try this one. What can you see now?
Perfect.
Is that
Perfect.
right?
We're good. Thank you.
So now you should see. Now it's not advancing to the next slide. How annoying. Okay, now it is. So see if this works. If there's central transmission, the same transmitters are released in the dorsal horn of the spinal cord, where they activate G-protein coupled receptors on second order spinal neurons. Can you see my proper slide now?
Yes.
But G-protein coupled receptors are not rigid, static on and off switches that sit at the plasma membrane of cells. Rather, they're better considered as dynamic signaling machines. We know from structural studies that receptors can adopt different conformations when bound to agonists, antagonists, or signaling partners. And this conformational dynamism can explain allosteric modulation, signaling bias, the effects of receptor oligomerization, or constitutive activity of receptors. Receptors also exhibit a positional dynamism in that upon activation, receptors can translocate from the plasma membrane to endosomes. Once viewed merely as a conduit for receptor trafficking to degradatory or recycling pathways, endosomes are now known to be an important site of signaling. We've explored the importance of this dynamic nature in the homeostatic control of pain and in drug discovery for analgesia. And I'd like to begin by discussing evidence that endosomes are platforms for signaling pain, and I'm going to focus my attention on the substance P neurokinin 1 receptor on second order spinal neurons. In many respects, this receptor is the poster child of failures in drug discovery to develop drugs to treat pain. From studies, usually in model systems, we have a good understanding of the dynamic properties of the Nk1 receptor. Upon binding substance P at the plasma membrane, the receptor becomes quickly phosphorylated by G-protein coupled receptor kinases and interacts with arrestins, which arrest or desensitize G-protein signaling at the plasma membrane. Arrestins are also adapter proteins for adapter protein 2 and Clathrin, and mediate dynamin-dependent receptor endocytosis. The receptor can continue to signal from endosomes by G-protein and arrestin-dependent mechanisms, which I'll describe. Sorting events in endosomes allow the receptor, freed from arrestins, to recycle and thereby resensitize, although after sustained activation, a portion of the receptor can be degraded. Now this, of course, is a picture of the Golden Gate Bridge, but you've perhaps not seen the bridge from this perspective. My wife took care of an iron worker on the Golden Gate Bridge who was in considerable pain, and as a reward for her good care, he gave her a pass to ascend to the top of one of these towers several hundred feet above the Pacific. Alas, she chose a typical San Francisco summer's day to make this excursion when the Golden Gate is shrouded in fog. I show this slide to illustrate that although we have a clear understanding of how receptors signal at the plasma membrane, signaling of receptors in endosomes is shrouded in a fog of uncertainty. We don't fully understand the mechanisms or the function, or know whether receptors in endosomes, rather than at the plasma membrane, might be a superior and valid therapeutic target. And these are the questions I'd like to address. To begin to study the dynamic nature of the Nk1 receptor, we used biophysical approaches to assess trafficking and signaling. The first was bioluminescence resonance energy transfer, or BRET, in which we express the neurokinin 1 receptor with a renylluciferase tag, and a partner protein such as KRAS, which is resident at the plasma membrane, or RAB5A, resident of early endosomes, tagged with Venus or YFP. Now, if these two proteins are within 10 nanometers, when we add the substrate, solentrazine, a BRET signal occurs and can be quantified. To assess signaling in subcellular compartments, we expressed genetically encoded FRET biosensors, including sensors for cyclic AMP, ERK, and protein kinase C. Now, these sensors can be reversibly modified, for example, by binding or dissociation of cyclic AMP, or by kinase or phosphatase activity. They're targeted to the plasma membrane, cytosol, or nucleus, allowing examination of signaling in subcellular compartments of living cells with a good spatial and temporal fidelity. And we've expressed these sensors both in HEK cells as well as in neurons. This is a BRET assay in HEK cells. Upon stimulation with substance P, there is a decrease in BRET between the Nk1 receptor and KRAS, and a corresponding increase in BRET between the receptor and RAB5A, consistent with endocytosis. At the 15-minute time point, treatment of cells with the Clathrin inhibitor PIT-STOP2, or the dynamin inhibitor, which was discovered in Australia and is called DINGO4A, inhibit removal of the receptor from the plasma membrane and its accumulation in endosomes, and inactive analogues have no effect. When we use a pharmacological tool, we use a genetic approach to confirm the result. An overexpression of dominant negative dynamin K44E also inhibited receptor endocytosis. To validate these results, we had examined the endocytosis of fluorescently labeled substance P. In vehicle-treated cells, substance P robustly internalizes after 30 minutes. Treatment with DINGO or PIT-STOP causes retention of the receptor at vesicles at or close to the plasma membrane, but the inactive analogues allow endocytosis to proceed, and therefore dynamin and Clathrin mediate substance P-evoked endocytosis of the receptor. This is a BRET assay, and here the sensor is confined to the FRET assay. Here the sensor is confined to the nucleus, and we're measuring nuclear ERK activity against time. In black, treatment with vehicle had no effect. In red, treatment with substance P resulted in a gradual and sustained increase in ERK activity, indicated by this representative image of a single cell. In blue, treatment with DINGO, the dynamin inhibitor, abolished the response, but inactive DINGO had no effect. And if we quantify the area under the curve in this 20-minute experiment, both DINGO and PIT-STOP abolish nuclear ERK activation. Downregulation of dynamin and Clathrin with siRNA have the same effect, and therefore Nk1 receptor endocytosis is necessary for nuclear ERK activation. We can also use BRET to examine the assembly of signaling complexes in endosomes. Here we used enhanced bystander BRET to examine BRET between mini-G proteins, these are engineered GTPase domains, tagged with renylluciferase, and either CAX, a resident protein of the plasma membrane, or RAB5A, of early endosomes in response to substance P. In cells expressing the full-length human Nk1 receptor, stimulation with substance P results in a rapid increase in BRET between CAX and G-alpha-Q, and also between Rab5A and G-alpha-Q, consistent with trafficking of the G protein to the plasma membrane and in endosomes. Nk1 receptor 311 is a naturally occurring truncated variant which does not internalize, and I'll talk more about that a little bit later. By using super resolution microscopy, we can detect the Nk1 receptor and G-alpha-Q in early endosomal antigen positive early endosomes. And the G-alpha-Q inhibitor UBO-QIC abolishes substance P-induced activation of nuclear ERK. So from these and many other experiments, we have a good understanding of how the receptor can signal in subcellular domains. The Nk1 receptor, of course, can signal at the plasma membrane. This generates cyclic AMP and protein kinase C in the vicinity of the plasma membrane and activates ERK, which translocate to the cytosol. But these signals are rapidly attenuated. The receptor internalizes by a dynamin and Clathrin dependent mechanism and can continue to signal from endosomes by mechanisms that include activation of ERK, which translocates to the nucleus. I showed you those results, and also activation of protein kinase C and cyclic AMP within the cytosol. We can block these signals using genetic and pharmacological approaches to disrupt dynamin or Clathrin and beta-arrestins. I didn't show those results. These results raise the question, what's the relevance of endocytosis and endosomal signaling for the control of complex physiological events in intact tissues or animals? To begin to address this question, we made cell-attached patch clamp recordings from Nk1 receptor positive neurons in superficial laminae of the rat spinal cord. The tissue was pre-treated with inhibitors of endocytosis, superfused with substance P for 2 minutes, and then action potential firing was measured over time. Treatment with substance P resulted in a rapid onset of firing that was sustained for approximately 10 minutes, as shown by the black dots here. Dingo inactive, the control compound, did not block action potential firing. Dingo inactive did not block the initial response, which presumably derives from the plasma membrane, but completely abolished the sustained response, as shown by the orange triangles in the orange bar here. Inhibitors of Nk1 and protein kinase C also abolished the sustained activation of firing, and therefore Nk1 receptor endosomal signaling is necessary for sustained substance P-induced activation of these neurons. We then examined the importance of Clathrin and dynamin for endocytosis and nociception in vivo. Inhibitors were injected intrathecally into conscious mice using a Hamilton syringe. When the syringe is in the right area, the tail flips. We then injected 5 microliters of a drug, here the endocytosis inhibitors, and returned the animal to its cage. 30 minutes later, the animal received an intraplantar injection of capsaicin, and we collected spinal cord to localize the Nk1 receptor by immunofluorescence and confocal microscopy. And at various times, nociception was assessed by measuring pore withdrawal responses to stimulation with calibrated von Frey filaments. This is an experiment in rats in which vehicle was injected into the spinal cord, and 10 minutes later, sections were collected. And this optical image of a single spinal neuron shows that the neurokinin 1 receptor is confined to the plasma membrane of the soma and neurites. 10 minutes after injection of capsaicin, there is a massive endocytosis of the Nk1 receptor that is attributable to the excitation of neurons and the release of substance P within the dorsal horn. After an intrathecal injection of inactive pit stop and then capsaicin as a control, the Nk1 receptor is massively internalized. But if we inject active pit stop, we can strongly inhibit internalization And that's seen very clearly in the single optical section. So if we take away the sections from this 3D projection, you can see the receptor is largely at the plasma membrane. We obtained identical results by using a dynamin inhibitor. And this is an assay of nociception. So in this graph, a downward deflection is an increased pain response. In black, in animals that had received an intrathecal injection of vehicle and an intraplantar injection of capsaicin, there is a sustained pain response. This can be partially inhibited by an intrathecal injection of a small molecule Nk1 receptor antagonist, SR14033. It's partly inhibited because other mediators like CGRP also contribute to the response. An intrathecal injection of pit stop or dingo strongly inhibited the response. I should point out that withdrawal responses of the contralateral paw were unchanged, and these inhibitors did not affect latency of the animals on a rotarod, so motor coordination appears to be intact. We confirm these results by injecting intrathecal dynamin-1 siRNA, which knocked down dynamin, determined by Western blotting of extracts of spinal cord at 24 and 48 hours, and also attenuated the nociceptive response to capsaicin. We were also able to reverse inflammatory pain induced by complete Freund's adjuvant injection into the paw. Injection of the endocytic inhibitors, dingo or pit stop, 36 hours after CFA, also reversed the pain response. Now, as the reviewers of our manuscript pointed out in quite exquisite detail, the difficulty of injecting inhibitors of dynamin and clathrin into animals is that these proteins regulate the trafficking of many receptors and ion channels. And therefore, we sought to identify a more selective approach to inhibit endocytosis of the Nk1 receptor. To do so, we focused our attention on the C-terminus of the Nk1 receptor, which is shown here. In humans, the Nk1 receptor has 407 amino acids. A naturally occurring variant is truncated at residue 312 and lacks this entire intracellular C-terminus. We expressed the full-length or truncated Nk1 receptor in HEC cells, along with beta-arrestin 2, and examined the effects of graded concentrations of substance P on recruitment of arrestins to the Nk1 receptor by using Brett. In cells expressing the wild-type receptor, substance P caused a concentration-dependent stimulation of Brett, but the truncated receptor was unable to recruit beta-arrestins. Whereas the wild-type receptor translocated to Rab5A positive endosomes in response to substance P, the truncated mutant remained at the plasma membrane and did not internalize. Now, the intracellular C-terminus of the Nk1 receptor contains a number of serine and threonine-rich regions, which might be sites of GRK phosphorylation and interaction with beta-arrestins. We synthesize these peptides along with a phosphopeptide or a scramble control peptide and conjugated them to the TAT sequence to allow membrane penetration and introduction into cells. In the arrestin-Brett assay, a mixture of these three Nk1 receptor peptides shown in blue strongly inhibited substance P-induced recruitment of arrestins to the full-length receptor where the control peptides did not. And in a cell surface ELISA assay, the mixture of the three peptides also blocked substance P-induced endocytosis of the receptor. When injected intrathecally into mice, the Nk1 receptor peptides inhibited capsaicin-evoked nociception and reversed CFA-evoked nociception, whereas the control peptides did not. Finally, we've used a genetic approach to assess the importance of endocytosis. We made knock-in mice expressing the full-length human Nk1 receptor or the truncated mutant. And in preliminary results, we found that an intrathecal injection of substance P caused a nociceptive response that was similar in wild-type animals in blue and in mice expressing the full-length human Nk1 receptor in black, but in animals expressing the truncated and internalization defective mutant, the substance P result is diminished, and this work is ongoing. So the observation that the Nk1 receptor is massively internalized after painful stimuli, and that the Nk1 receptor can continue to signal from endosomes to regulate neuronal excitability, raised two questions. Can we develop strategies that will selectively antagonize receptors in endosomes, and are receptors in endosomes a viable therapeutic target? Well, I'll move on to the second part of my talk, which is therapeutic targeting of endosomal receptors. We've used several approaches. The first I've already described, and that's to use inhibitors of endocytosis. The second approach is to use the structure of the Nk1 receptor to design endosomally targeted or biased ligands that might preferentially interact with conformations of receptor in acidified endosomes compared to conformations of the plasma membrane. The third approach has been to conjugate antagonists to lipids, which would escort the antagonist to endosomes and retain it in endosomes for prolonged periods. And the final approach has been to encapsulate antagonists into stimulus-responsive nanoparticles designed to enter endosomes and release their cargo in the acidified endosomal environment. I'll briefly discuss endosomally biased antagonists. Well, the structure of the Nk1 receptor is available in association with a variety of antagonists, and we could use this structure to design ligands that might preferentially stay bound to the receptor in the acidified endosomal environment. There is precedent for this approach, as the Stein laboratory has published in Science quite recently an analog of fentanyl known as NFEP. It's a fluorinated analog which preferentially binds the mu-opioid receptor in the acidified extracellular environment of inflamed or damaged tissue. We've obtained NFEP and found, for example, in this assay in HEC cells expressing the mu-opioid receptor, that it more powerfully inhibits cyclic AMP production at an extracellular pH of 6.5, but has no effect of pH 7.4. To examine pH dependence of inhibition of neuronal excitability, we made patch clamp recordings from mouse dorsal root ganglion neurons, exposed the cells to NFEP at pH 6.5 or 7.4, washed and then measured RIA base, the minimum current required to fire an action potential. An increase in RIA base would denote a decrease in excitability. And whereas NFEP increased RIA base at pH 6.5, it did not at pH 7.4. We're currently pursuing this approach to develop superior antagonists of the Nk1 receptor. The third approach is to develop tripartite lipidated compounds or probes. They have three components: cholestanol, which would span a membrane, or ethyl ester control, which would not insert into a membrane, a PEG-12 linker, and then a cargo. And the cargo could be a cyanine 5 dye if we want to examine the trafficking of these probes throughout the cell, or it could be a first-generation peptidic antagonist, such as spantide, or a small molecule antagonist, such as the neurokinin 1 receptor. And we can use computer modeling to understand the optimum PEG-12 link or PEG linker, which would allow presentation of antagonists to binding sites for antagonists on receptors in transmembrane domains. In this experiment, we incubated HEC cells with a cyanine 5 ethyl ester control probe or a cyanine 5 cholestanol probe and examined the uptake of the probe into cells. The probe is added at time 0, and the ethyl ester probe remains extracellular and is not taken up into the cells. In contrast, when we add a cholestanol conjugated probe, it's immediately incorporated into the plasma membrane and then within minutes accumulates in endosomes. With time, we can see that the probe accumulates in endosomes, in this experiment also containing the Nk1 receptor tagged with green fluorescent protein. And here we used a probe, it's a spantide cholestanol probe, and the spantide itself is labeled with cyanine 5. To examine whether this tripartite antagonists can block endosomal signaling, we first measured ERK activation. The protocol was to pre-incubate cells with antagonist, wash, wait for 4 hours to allow the antagonist to accumulate in endosomes, and then examine substance P-induced ERK activation with a FRET biosensor. In red, substance P stimulates ERK activation. This was unaffected by conventional unconjugated spantide or by a small molecule antagonist, but was abolished by spantide conjugated to cholestanol. We then examined the effects of these probes on action potential firing, again using this pre-treatment protocol. Spantide did not block signaling. Spantide cholestanol blocked the effects of substance P, did not block the effects of substance P on the initial firing, which likely derives from plasma membrane receptors, but completely attenuated the sustained response. Finally, the probes were injected intrathecally into animals, in this experiment 3 hours before an intraplantar injection of capsaicin. Here, unconjugated spantide or the small molecule antagonists were unable to attenuate capsaicin about nociception, but spantide cholestanol strongly inhibited the response. And these tripartite antagonists were also capable of reversing inflammatory pain induced by complete Freund's adjuvant. The final approach to manipulating the endosomal receptor has been to develop stimulus-responsive or pH-tunable nanoparticles. The nanoparticles are made of monomers. There is a hydrophilic shell of monomers designed to promote circulating half-life and cellular uptake. A hydrophobic core of a monomer which was designed to reduce cellular toxicity, and a dipmer monomer to promote pH-dependent disassembly. Now, the key feature of this monomer is that it has a pKa of 6.1. So at acidic endosomal pH, the monomer would be protonated, and like charge repulsion would cause disassembly of the nanoparticle and release of its cargo. As a control, we made a stable nanoparticle with the same hydrophilic shell, but with a core of BMA, which would not be protonated, and therefore this would not disassemble. And into the core of the particle, we've incorporated a number of hydrophobic dyes, but we've also incorporated a hydrophobic small molecule Nk1 receptor antagonist, Apripitan, which is clinically used to treat nausea and vomiting, but which failed in trials against chronic neurological diseases and including chronic pain. The concept is that these monomers would assemble in an aqueous environment and encapsulate within them a hydrophobic cargo, and that this process would be reversed in an acidified endosome. Transmission electron microscopy reveals that the particles are spherical and uniform. They have a diameter between 30 to 40 nanometers, a small negative surface charge, and we're able to obtain over 50% Apripitan loading. To examine pH-dependent disassembly of the particles in a test tube, we filled them with the dye Nile red. So the important thing to remember about Nile red is that it fluoresces in the hydrophobic core, but is quenched in an aqueous environment. And so a loss of fluorescence would be attributable to particle disassembly. As we reduce the pH below 6, fluorescence of the BMA stable particle is unaffected, but fluorescence of the dipmer pH-tunable nanoparticle declines, indicating particle disassembly at pH less than 6. To examine disassembly in cells, we filled the particles with a different dye. This is coumarin 58. It's the opposite of Nile red. This doesn't fluoresce within the particle, but only fluoresces in the aqueous environment. So if the cell goes green, the particle's disassembling. So we added the particles to hex cells at 0 minutes, and you can see within a couple of minutes, the cells begin to go green, and they become very green after about 10 to 20 minutes, indicative of particle disassembly and dye release. At the 30-minute time point, treatment of cells with a Clathrin inhibitor pit stop or a dynamin inhibitor dingo strongly inhibits the fluorescence. Treatment with ammonium chloride or with baflomycin A, an inhibitor of vacuolar hydrogen ATPases, these treatments would block vesicular acidification, also blocked the fluorescence, and we quantified the data here. Here is the control, and these are the inhibitors showing a very large inhibition by pit stop, the Clathrin inhibitor. So the particles have to be taken up by dynamin and Clathrin-dependent endocytosis and require endosomal acidification to disassemble. We've filled the particles with a fluorescent dye, cyanine 5, and in HEC cells expressing the Nk1 receptor tagged with green fluorescent protein, we can see that the particles distribute to endosomes which contain the neurokinin 1 receptor. We first assessed the ability of DIPMA aprepitant to inhibit endosomally mediated nuclear ERK signaling, and found that whereas free aprepitant in blue had no effect on the substance P response, DIPMA aprepitant completely abolished the response. In the assay of action potential firing in slices of rat spinal cord, aprepitant or the stable particle were unable to block the sustained effect of substance P, whereas DIPMA aprepitant abolished the sustained effect shown here. When injected intrathecally into animals, the cyanine 5 DIPMA particles are retained at the site of injection for up to 1 day. If we collected spinal cords from these animals, you can see that the cyanine 5 labeled particles are found in laminae 1, 2, and 3, as shown in higher power. And some of these particles are taken up by neurons identified by the neuronal marker PGP 9.5. This three-dimensional projection shows accumulation of the particles in many cells in laminae 1 of the spinal cord, shown here. At various time points, 1 and 4 hours after injection, we use mass spectrometry to quantify the retention of aprepitant within the spinal cord. And in animals treated with aprepitant in the pH tunable or stable particles at both time points, there is retention of aprepitant, whereas free aprepitant is rapidly removed and is hardly detected after 4 hours. In this experiment, nanoparticles or free aprepitant were injected half an hour before an intraplantar injection of capsaicin, and then capsaicin-evoked nociception was assessed. In black, animals receiving capsaicin alone have this sustained nociceptive response for up to 4 hours. In gray, the empty particles as a control had no effect. The blue triangles show that free aprepitant had a small inhibitory effect, as did aprepitant in the non-dissembling BMA particle, whereas in red, aprepitant in the pH tunable DIPMA particle had a larger and far more sustained effect. At the 4-hour time point, we collected spinal cord and stained for phospho-ERK as an index of neuronal activation. And we saw that the DIPMA aprepitant completely abolished ERK activation, whereas the free aprepitant was far less effective. In a model of inflammatory pain induced by complete Freund's adjuvant, when injected 48 hours after CFA, we see that free aprepitant or the stable particle have a small effect, whereas the DIPMA aprepitant has a larger and more sustained effect. And then in 2 models of neuropathic pain in both rats and mice, the DIPMA particles shown here in pink or here in red are far more effective than the free aprepitant or the stable particles at all time points. It's a larger effect and it's more sustained, and is far more sustained than the analgesic effect of systemic morphine in this experiment. So to summarize, this video emphasizes the importance of endosomal signaling of pain. Substance P binds to the Nk1 receptor at the plasma membrane. It's quickly phosphorylated by G protein coupled receptor kinases and then associated with beta-arrestins. Beta-arrestins are adapter proteins for adapter protein 2, which is coming up here, and also for Clathrin, which together mediate endocytosis. Substance P continues to signal with the Nk1 receptor in endosomes by G protein and arrestin-dependent mechanisms, which activate ERK. ERK translocates to the nucleus and also activates protein kinase C and cyclic AMP, which are retained in the cytosol. And together, these events are responsible for sustained neuronal excitation and pain transmission. Well, what's the possible relevance of endosomal signaling to painful human disorders? In this last part of my presentation, I want to discuss a different G protein coupled receptor, protease activated receptor 2 or PAR2, which is expressed in a subset of nociceptors and can be activated by a variety of proteases which are generated in diseased and damaged tissue. Proteases and PAR2 have been strongly implicated in the painful human disorder of irritable bowel syndrome, or IBS. And evidence for this proposal largely derives from analysis of mucosal biopsies from either patients with IBS or healthy control subjects. And colonic mucosal biopsies are collected from IBS patients as a routine diagnostic tool. Biopsies from patients with IBS contain a larger number of tryptase positive mast cells, which are often in close proximity to nerve fibers that might express PAR2, compared to biopsies of control colonic mucosa. The IBS biopsies release the PAR2 activating enzyme tryptase in far higher amounts than healthy control biopsies. When supernatants from IBS or control biopsies are instilled into the colonic lumen of mice, there is an enhanced visceromotor response recorded by an EMG measurement of abdominal muscles to colorectal distension with a biorostat, then after infusion of control supernatants. And the response to the IBS supernatants in wild-type animals is diminished in PAR2 knockout animals. Well, how do proteases activate PAR2? The canonical mechanism by which tryptase and trypsin activate PAR2 involves cleavage at this arginine-serine site within the extracellular tail of the receptor to expose a tethered ligand shown in green, which then binds to and activates the cleaved receptor. After activation by trypsin or tryptase, PAR2 couples to G proteins, particularly G-alpha-Q. It also recruits beta-arrestins, robustly internalizes, and can continue to signal from endosomes to activate MAP kinases. A number of other proteases, including serine and cysteine proteases, cleave PAR2 at distinctly different sites. They include elastase, cathepsin S, and legumine. After activation by these proteases, PAR2 couples to different G proteins, in particular G-alpha-S and G-alpha-12-13. But importantly, it doesn't recruit beta-arrestins and does not internalize. Instead, it signals by bias mechanisms that operate almost exclusively from the plasma membrane rather than endosomes. To begin to examine the tissue distribution and the trafficking of PAR2 in vivo, we generated knock-in mice in which PAR2 was fused at the C-terminus to monomeric ultrastable green fluorescent protein. Analysis of these animals demonstrated a very high expression of PAR2 in epithelial cells, including keratinocytes, and here colonocytes, where PAR2 GFP is found predominantly at the basolateral membrane. However, after inducing colitis, there is a massive redistribution of PAR2 from the plasma membrane to endosomes, which is attributable to the activation of trypsin or tryptase-like enzymes as a result of inflammation. PAR2 GFP can also be detected at the plasma membrane of dorsal root ganglion neurons. Here in culture, exposure to trypsin results in a massive internalization of the receptor, quantified here as the cytosolic to plasma membrane ratio. In contrast, exposure to elastase or cathepsin S did not evoke endocytosis, and trypsin-evoked endocytosis is prevented by dingo, but not by dingo inactive. To determine whether PAR2 in epithelial cells or neurons is responsible for PAR2 evoked or for protease evoked pain, we deleted PAR2 in NAV 1.8 positive neurons and confirmed deletion of PAR2 by microscopy and also by functional calcium imaging of cultured cells. Here we go from 20% responsive neurons to around 7% in the PAR2 NAV 1.8 mice. An intraplantar injection of trypsin or elastase into wild-type animals in black induces a sustained von Frey filament response that lasts for 3 hours. However, in the PAR2 NAV 1.8 mice, the sustained response is completely attenuated, indicating that the presence of PAR2 in NAV 1.8 nociceptors is necessary for the nociceptive response to these proteases. We then investigated the role of endocytosis in pain by injecting endocytosis inhibitors 30 minutes before an intraplantar injection of proteases. Injection of dingo, shown in the brown filled triangles, attenuated the nociceptive response to trypsin, but not to elastase, consistent with the ability of trypsin, but not elastase to evoke endocytosis. To examine mechanisms of protease-evoked sensitization of nociceptors, we made patch clamp recordings from primary cultures of mouse dorsal root ganglion neurons. The tissue was briefly incubated with proteases, washed, and then reabase was measured immediately after washing or 30 minutes later. Exposure of cells to trypsin resulted in a decrease in reabase, in other words, an increase in excitability that was detected immediately after washing and was sustained for 30 minutes. The dynamin inhibitor dingo did not affect the initial response, which requires plasma membrane signaling, but reversed the sustained response, and pooled data are shown here. A MEK inhibitor also prevented the sustained response, and this effect was abolished by a PAR2 antagonist. In contrast, neutrophil elastase also causes an initial and sustained increase in excitability of neurons, but this is completely unaffected by an endocytic inhibitor, consistent with the inability of elastase to evoke endocytosis of the receptor. Well, could endosomal signaling contribute to the effects of proteases released from IBS biopsies on the pain response? Well, to examine this question, we collected biopsies from the colonic mucosa of healthy control subjects or subjects with irritable bowel syndrome, incubated in media, and then collected supernatant. Neurons were exposed to supernatant, washed, and reabase was measured 30 minutes later. Compared to healthy control subjects, tissues or neurons treated with IBS-D supernatant showed a decrease in reabase, which could be inhibited by a PAR2 antagonist. And pooled data are shown here. This response was also inhibited by dingo 4A, whereas dingo had no effect in the healthy control subjects. We sought to selectively antagonize PAR2 in endosomes, and to do so, we generated a tripartite antagonist in which the small molecule inhibitor of PAR2, I343, was conjugated to PEG12 and cholestanol. A cyanine 5 labeled tripartite probe was taken up into cultured dorsal root ganglion neurons and co-localized with PAR2 GFP, so we can deliver an antagonist to endosomes of DRG neurons containing PAR2. The PAR2 antagonist MIPS 15479, selectively inhibited the sustained hyperexcitability induced by trypsin, and also completely inhibited the response of IBS-D supernatant, but had no effect in the control. So to summarize, proteases that activate PAR2 by canonical and bias mechanisms can sensitize nociceptors through signaling at the plasma membrane or in endosomes. After stimulation with trypsin or tryptase, PAR2 can initially signal from the plasma membrane and sensitize nociceptors by a PKC-dependent mechanism. But with time, the receptor internalizes and continues to signal from endosomes and sensitizes neurons by an ERK-dependent pathway. Conversely, after activation by cathepsin S, elastase, or legumain, the receptor signals from the plasma membrane to mediate sensitization. I've described mechanisms by which substance P signals from endosomes to regulate excitability of spinal neurons, and which PAR2 signals from endosomes to regulate excitability of primary sensory neurons. We've also reported that the calcitonin gene-related peptide receptor signals from spinal neurons to regulate nociception, and we're currently exploring the role of endosomal signaling in migraine, and CGRP is a validated target for the treatment of migraine. And finally, we've recently obtained evidence that the opioid receptor, which of course inhibits pain transmission, can signal from endosomes of spinal neurons and primary sensory neurons. And this evidence was obtained by generating a different type of particle with a shell of targeting groups, allowing selective targeting of the particle to neurons expressing the delta opioid receptor, and a core of a delta opioid receptor agonist that would be gradually released under the acidic and reducing conditions of endosomes. Our results then suggest that endosomes can generate, endosomal receptors can generate sustained signals in subcellular compartments that underlie important pathophysiological processes such as pain transmission. That targeting receptors in endosomes is a new approach for drug delivery, which may have broad implications because many G protein-coupled receptors internalize upon activation, and GPCRs are the target of 30% of clinically used drugs. And our results also raise the possibility that the ineffectiveness of GRP-directed drugs in clinical trials might reflect their inability to effectively target and engage conformations of receptors in endosomes in chronic disease states. And I'd like to close by thanking my collaborators at NYU, at Columbia, and at Queen's University, as well as collaborators in Europe and Australia, to acknowledge funding from the NIH, the Department of Defense, the Australian National Health and Medical Research Council, and Australia Research Council, and to disclose collaboration with Takeda and Endosome Therapeutics. And thank you for your attention.
That was terrific, Nigel. Thank you.
Thank you.
I'm not going to take the opportunity to ask all my questions first. I will cite a couple of the questions. So Stefan Friedrichsdorf is, it's actually, so here's a clinician, a pain clinician, who obviously is aware that the Nk1 receptor antagonists have failed miserably in these traditional ones, except they're effective in Emesis.
Yes.
And it's clear that you're implying that these might be effective in patients compared to Apripitan. And when he doesn't ask the question, but are you anticipating this? How long might it be? And more importantly, I think, is how might it be delivered?
So, that is correct. We do anticipate that encapsulating antagonists into pH-tunable nanoparticles would enhance their therapeutic efficacy. And we have a collaboration with NCATs, who, if work goes according to plan, could take particles containing Apripitan or similar drugs all the way up to an IND. I think the attractiveness about using nanoparticles is that you can encapsulate into them multiple antagonists of GPCRs and thereby could circumvent the inherent redundancy in the pain pathways, which of course is important, and so it's mediated by a lot of different transmitters. It's also possible, and I didn't really get into this, to target the particles selectively to neurons by decorating the shell with targeting groups. And so we've just got a HEAL grant to do that, where we are coating them with opioids and putting into the core Nk1 and CGRP antagonists. So the route of delivery, Alan, would depend on the target. If we're targeting central processes in the spinal cord, it would involve an intrathecal injection, because after systemic delivery, I doubt these would get centrally. However, if you're targeting a peripheral mechanism, perhaps PAR2, a peripheral route might be, well, would be appropriate. So I think it would depend upon the receptor you're trying to target.
Okay. And there's a question, well, first a comment from Aaron, who said it was a wonderful talk. Really appreciated the suite of new tools that you've described. And then he goes on to say, and I'll quote, generally GPCR C-term helix 8 mutants and truncations do not fold properly and make it to the surface. So he's wondering if it's known if there is an exception for the Nk1 receptor.
Well, that's a great topic and a great question, because we're currently looking at that. So we've been expressing the truncated Nk1 receptor, or the full-length receptor, and we're using mini G proteins to look at recruitment of different G proteins and arrestins to various compartments by Brett. So what I can say is that the truncated receptor is expressed at the plasma membrane, but at a considerably lower level than the wild type receptor. I couldn't put a number on it. However, in the mice, which are expressing the truncated receptor, we do see that substance P can cause an inflammatory response, and it can also cause a nociceptive response. But it's not as clear as I thought. I thought that when I made these mice, that it would allow me to provide genetic evidence that internalization is important for pain transmission, but it's more complicated, because I agree with you. I don't think the truncated receptor is completely trafficked normally to the plasma membrane.
So I had a couple of questions, if I may. I believe that you're making the case that the action against capsaicin was against the kind of prolongation of the behavior of the sensitization, because I think most of your endpoints were in response to, say, von Frey. The other side of the coin is capsaicin itself produces a lot of behavior. And I know that if you kill the Nk1 receptor expressing cells in the cord like Manti did,
yes,
with substance P supportin, you don't eliminate the response to capsaicin. What you do eliminate is, or significantly reduce, is the sensitization.
Yes.
Is your treatment in the animals affecting the actual response to the capsaicin itself?
The
animals will lick the paw immediately.
Yeah. You know, we have looked at we didn't measure licking or nociceptive behavior. We did look at nociceptive behavior to formalin, and we could see an inhibition of both phases of the formalin response with, for example, inhibitors of endocytosis or the lipidated spantide. But we didn't do that experiment with capsaicin.
Okay. The other question is more a little esoteric on my interest. There's another one from, I'll come back to Joe in a second, but it's just, I happen to, I'm really impressed with the IBS model, and I'm kind of curious, and you're probably aware that IBS manifests as two very different forms, one with constipation, one without constipation. And the former is incredibly sensitive to the guanylate cyclase inhibitor, linaclotide.
Yes.
And I'm kind of curious whether the treatment that you manifest really had any relationship to the difference between those two conditions?
Yeah, so we looked at diarrhea predominant IBS patients. So all the work that I was presented was from IBS-D. If I know that most of this work came from Natalie Vignol's lab and Steve Banner's lab. And I know in her original paper, Natalie, I'm almost certain, looked at IBS-C and IBS-D. And I think showed a difference, and that's why we focused on IBS-D, but I'm not 100% sure. I mean, IBS is a very complex and poorly understood disorder, and not only is it constipation or diarrhea predominant, it can be alternating, and there are forms of IBS which have been related to a post-infectious state or a post-inflammatory state. It's difficult to study. We've relied mostly on using biopsies. We have done a little bit of work in mouse models where you induce an inflammation. You can induce colitis in mice, let the colitis resolve, and then a month later, when there's no histological sign of inflammation, the animals still show a sensitivity.
Okay. Joël Braz asked the following question. PAR2 is expressed not only in sensory neurons, but of course also in immune cells, in the skin, probably extensively. And the question is whether, well, do you have a sense of the relative contribution of non-neuronal PAR2 vis-a-vis the endosomal functional inhibition in the contribution of the enzyme to pain?
Yeah, so you're right. So PAR2 is most highly expressed in epithelial tissues. In the skin, it's highly expressed. It's found in colonocytes, enterocytes, and it's expressed in a small subset of neurons in mice. It's higher in rats, but it's expressed at lower levels. So when we deleted PAR2 in NAV1.8 positive neurons and injected a protease into the pore, there was an initial nociception, which we could detect at 30 minutes, which was the same in wild-type animals and in mice lacking PAR2 in NAV1.8 positive neurons. So presumably that response it is perhaps mediated by PAR2 in keratinocytes. But the sustained response was lacking in mice that were lacking PAR2 in NAV1.8 positive neurons. So that response presumably requires PAR2 in neurons. Mm-hmm. So in answer to your question about could endosomal signaling, I think this is a question, in epithelial cells play a role. We're currently investigating that. And we have evidence that actually from biopsies of human colon. So we take biopsies of human colon and incubate them with PAR2 agonists and look at cytokine release. And you can inhibit protease-evoked cytokine release by inhibitors of endocytosis. And I think those cytokines are coming from colonocytes. So I believe that endosomal signaling plays a role in that inflammatory response you see in colonocytes. We haven't published that, but that's what the results are showing so far.
I'm hesitant to take over all these questions, but the other question that I've always wondered about is in Emesis. Do you have an explanation, any idea why Apripitan is so effective in Emesis, where presumably the afferents, the no-dose afferents, if that's what's being affected, are releasing glutamate and substance P, yep. And it must be a pure membrane effect, but not effective for pain?
Yeah, I've been asked that question before, and I've thought about it, and I have to speak to somebody about the timing of administration of Apripitan in patients. I mean, and I just don't know. Do patients get Apripitan before the chemotherapeutic agent? That's a possibility, because then if you block the receptor at the cell surface, you block any subsequent signaling completely.
Well, I can speak specifically because my late wife took it, and it was certainly after, because they wait to find out if
Yeah.
If he was nauseous. They didn't treat ahead of time.
Yeah.
But I don't know what clinically, how it's usually done.
Yeah, and I don't know the answer to that. All I can do is really speculate. And I mean, obviously it would suggest that this receptor can perhaps signal from the cell surface to mediate the emetic effects, and aprepitant can effectively block it there. It may be in this population of neurons that the receptor doesn't internalize or is very rapidly recycled. And I just don't know. I think the striking thing in a pain state is, if you look in animals with inflammatory pain or with neuropathic pain, which has been going on for weeks, there's just a profound redistribution of the receptor from the cell surface to endosomes. Now, I think you're just changing the steady state distribution. Normally, under steady state, it's at the plasma membrane. In a pain state, presumably due to continuous release of substance P, it's internalized. I think it's probably always going round and round, but you just, in other words, recycling, but you just change the steady state distribution, and perhaps you get accumulation of substance P in endosomes. So I've not really answered your question, Alan, but I think it's the best I can do.
There's a question from Tia, who asks, do you think that in the clinic, you'd have to actually block both the plasma membrane and the endosomal signaling with, say, you might combine both caged and free apripitant? Was there any value to that, or would the caged apripitant be sufficient?
Well, it's a very difficult question to answer, because it's very challenging to selectively inhibit one and not the other. Mm-hmm. But even if we deliver aprepitant in a pH tunable nanoparticle, our evidence suggests that this particle will only release its cargo in a cell. So you would block the endosomal receptor, but of course aprepitant could diffuse out of the cell and block the receptor at the cell surface. Right. We have no way of controlling that. What I think is clear is that if you, the way I see it is that putting it into a nanoparticle is like giving an infusion of the drug into the endosomal network. And that certainly maintains aprepitant at higher concentrations in the cord. We showed that by extracting cords from mice and quantifying aprepitant with mass spec. So I believe maintaining this high concentration of drug within the endosome is therapeutically beneficial. But it may be that blocking the receptor at the cell surface would be useful as well. You know, we might be able to get at that question if we can develop antagonists which show a big preference for binding at acidic conditions versus neutral conditions, like Christoph Stein's fentanyl analog.
Okay. I don't see any more questions. I'll ask one last one, which for the pain basic scientists who, the big push these days, of course, is to try to get the mouse or the rat to tell you that something bothers them rather than a reflex. And so I'm curious whether you have seen, because the lamina 1 cells that you're affecting are sending the message to the brain to drive the experience of pain. Yes. They're not generating the reflex, or they may be contributing to it. And to what, have you done studies, the classic place preference aversion, to get a handle on whether the cortex is being affected? Yeah, so we have done that. So we've looked at cold allodynia, which is the same as the VFF response, and that the nanoparticle encapsulated drug is far more effective. We've also looked at spontaneous behavior of the animals. So we call it the box, but it's a box with multiple lasers in, and it allows us to carefully monitor animal behavior. And it's ongoing work, but the preliminary results suggest that when we use nanoparticle encapsulated aprepitant, it's far more effective at reverting the abnormal behavior in mice with neuropathic pain than free aprepitant. But it's ongoing, right. So obviously it's an important thing to do. And Michael Groper added the point that the Apripitan works better when administered prior to chemotherapy.
Oh, that's really good to know. Thank you, Michael.
Um,
Which would make sense because you block the initiation of signaling at the cell surface, and you would also block endocytosis and endosomal signaling. If you don't activate the receptor
right.
at the plasma membrane, it will never get in.
Well, I'm sure that Mark will express his thanks, but I would like to, on behalf of everybody, I don't know what the honorarium is for the person who handles questions, but I'll check with...
Wait a minute.
Nice try, Alan.
But
We'll
really,
take that
thank
under
you so much,
consideration.
Nigel. It was a great kickoff to PARC, a terrific kickoff.
Thank you.
Turn it over to you.
Yeah.
Pleasure to see you all again.
And yes, thank you again, Nigel, and all the participants. And Alan, thanks again. All right. Have a nice afternoon.
Thank you. You too. Bye-bye.
Cheers.
Safe travels to get home, Nigel.
Yes, thank you.